This is a model of the ventricle system of the brain. If you'll remember, the entire center of the central nervous system is hollow, and this represents part of it. If you had injected latex into the ventricular system and then removed the neural tissue from around it, this is what you would end up with. So we have two paired lateral ventricles. These are these long, almost C-shaped structures. These lie under the cerebral cortex within the telencephalon. And underneath them, I'm going to turn this back. And by the way, this is anterior and this is posterior. I'm going to be flipping this around as we look at this several different times. Right now, you are looking at this as though you were looking at the underside of the brain. So the eyes would be located here, and the rest of the body would come out towards the camera. From the inferior view, you can see some pink-colored regions within those ventricles. That pink coloration represents choroid plexus, which is the vascular structure within the ventricular system that produces cerebrospinal fluid. This structure is made of a capillary bed that is supported by pia mater and covered with ependymal cells. And as serum squeezes out of the capillary bed, the ependymal cells ensure that the constituency of the cerebrospinal fluid is appropriate for keeping the neurons happy. All right, so we have, again, the lateral ventricles here and here. In between them is a sheet of neural tissue called the septum pellucidum. It lies right between them and separates them. When we move down, we move into another region of the ventricular system called the third ventricle. This is all the third ventricle, and there are two connecting canals between the lateral ventricles here and this more central third ventricle. These are called the Interventricular Foramen of Monro. In the third ventricle, you can see an area, it almost looks like the eye of a rooster. This is an area that represents where there isn't any ventricle. This is actually a mass of neural tissue that is present in some individuals, but not in all of them, that connects the paired thalami. So there is a thalamus, which is a nucleus, a cluster of nuclei here, and a thalamus that would be found here. The interthalamic adhesion or mass penetrates through and goes right through the middle of the third ventricle. Down below, this is also part of the third ventricle, but what forms the lateral walls around this lower portion of the third ventricle is the hypothalamus. So you would find the thalami here and here paired, and then the hypothalamus is also a paired structure that would form the lateral walls of the third ventricle. As we move posteriorly and inferiorly, we move through the cerebral aqueduct, which runs right through the midbrain and connects the lower ventricle, this is the fourth ventricle, with the third ventricle more superiorly. This fourth ventricle lies anterior to the cerebellum and behind the pons. You can see as I roll this over for a posterior look, more choroid plexus down in the fourth ventricle, and there is also choroid plexus present in the third ventricle. One last thing I want to point out is that all of this cerebrospinal fluid that's being produced has to get out into the subarachnoid space, and it exits the ventricular system through holes, 3 holes, in the fourth ventricle. There are 2 lateral apertures that are located at about this point, and then inferiorly there is a third aperture called the median aperture, which also permits CSF to go into the subarachnoid space.